In our last video, we modeled the dance of meiosis, how diploid cells divide into 4 unique haploid cells. But now comes the big question. Which one of these cells actually becomes the next generation? In this video, we're putting the laws of probability to the test to see exactly how fertilization works and what our simulated baby will actually look like. Let's talk about the fate of the gametes. So in these 4 piles, we have 4 haploid cells that were created in the simulation. First, let's take a look at the difference between parents. If you carried out the simulation using chromosomes from the male parent, all 4 haploid cells produced our viable sperm. Any one of these piles could represent the sperm that fertilizes the egg. But in the female simulation, it works a little bit differently. While meiosis produces 4 cells, they aren't created equally. Only one of these piles becomes the functional egg. What happens to the other 3? They're called polar bodies. They eventually break down and are reabsorbed by the body. To keep this scientifically accurate, we're going to use a spinner. For our first spin, we'll select which of the 4 female cells becomes the egg. Spin 2 selects which swimmer wins the race to the egg. The haploid egg and the haploid sperm come together to form a diploid zygote. This process is called fertilization, the moment a unique set of genetic instructions gets locked in. Let's talk about the math of this process. You might wonder if we could get the same baby twice. In our simulation, we are using 7 pairs of chromosomes. Because of independent assortment, the number of different genetic combinations possible in our simulation is 2 to the n, where n is the number of chromosomes. Calculating 2 to the 7th, we get an answer of 128. When we carry out the process in male or female cells, we have 128 different possible outcomes. If you multiply the possibilities of the egg by the possibilities of the sperm, we see that we get a result of 16,384 possible genetic outcomes. And that's without even factoring in the process of crossing over, which wasn't used in this simulation. This is why siblings look similar, but are never identical, unless they're monozygotic twins, that is. Now let's analyze our results. I'm going to analyze these 2 sets of chromosomes. The pink chromosomes represent the egg, the blue chromosomes represent the sperm. As they come together during fertilization, we form a diploid zygote. If our zygote gets 2 X chromosomes, it will become a biological female. If the zygote receives 1 X and 1 Y chromosome, it will become a biological male. I can see here that I have an X chromosome from the male parent, and we have an X chromosome that came from the egg. So this zygote will develop into a female baby. Next, we look at the genotypes. By looking at the alleles on each chromosome pair, we can determine the phenotype. So genotype means which alleles did the zygote receive. The phenotype will be the observable trait that individual would express. We've gone from meiosis to fertilization, proving that biology is really just a mix of complex machinery and a little bit of randomness mixed in. If you'd like to learn more about the chromosomal basis of inheritance, you might want to check out this video right here.